Hi everyone, and welcome to the first lecture on energy flow and food webs. The learning objectives for this module are for you to be able to firstly define and provide examples, the main ways living organisms obtain energy, and secondly, how energy is transferred among biotic components of an ecosystem. So today's lecture will relate back to some of the concepts we discussed in the first lecture. However, then we focused on the decomposers and the energy flow there. Here we're going to put the information into the context of in the entire trophic pyramid in food web. First of all, we're going to start with a little bit recap on some basic energy principles. Essentially, energy is the most basic requirement for all living organisms, and it's used to power biological processes for both activity and growth. So an important point about energy that you need to remember is that there's the first law of thermodynamics. That is, that energy can be transformed, but it cannot be created or destroyed. Now, hopefully you'll also remember that energy is driven to drive biological processes comes from either light, which we'll come back to in a moment, or from chemicals, where the basic principle is that energy can be gained from an atom by pulling that electron away from the protons. This electron movement, if you remember, occurs during reduction and oxidation processes, or redox reactions. In this case, if you recall from your high school textbooks, here an atom can become reduced when it undergoes a chemical reaction in which it gains electrons. That is, it is the acceptor in a redox reaction. And an atom then becomes oxidized when it undergoes a chemical reaction in which it loses electrons. That is, it becomes the electron donor. If the energy released by the atom that loses an electron is more than the energy that is used to add an electron to an electron acceptor, then the remaining energy can be used for other biological processes. So, how does this fit into ecology? Well, all living organisms can be grouped into their nutritional classes based on how they satisfy their energy and carbon requirements. So, there are two broad categories. One is the autotrophs, shown here. There's a picture of a bunch of them here, and they include organisms such as plants and algae and several bacteria and archaea. These organisms can use inorganic carbon dioxide as the carbon source and make their own organic molecules by reducing that carbon dioxide into things like glucose. Autotrophs can then be broken down themselves into the ways in which they can carry out in which they gain the energy to carry out those reactions. If they gain their energy from light, they can be characterized as photoautotrophs, and they can use special light-trapping photosynthetic pigments to capture that energy from light photons. You're all going to be familiar with the major chlorophyll pigments found in plants, but light-harvesting pigments are not all just green. And in fact, for example, here on your slide, you can see a bunch of flasks containing cultures of different phototrophic bacteria, and each of them having different pigments. So the diversity of these pigments allows these microbes to capture light at different wavelengths, allowing those organisms to essentially occupy and proliferate in different and specific niches based on the light availability. So let's put that energy captured by these autotrophs into the context of energy flow. This is where the terms such as gross and net primary production become relevant. Take a look here at this schematic showing a representation of energy flow from primary productivity from the plants here in the schematic. About 1% of the energy from the sun that hits the leaves is absorbed and used for photosynthesis, thereby contributing to gross primary production. This is the rate primary producers can fix that CO2 and turn it into glucose. And it's standardly measured as units of carbon per unit area per unit of time. For example, it could be grams of carbon per square meter per year. Now, only about 30 to 50% of that fixed carbon by the plant is then lost by respiration. And so it becomes unavailable to consumers. The rest then goes on for things such as growth and reproduction, nutrient storage, and defense strategies that the plant has to live with. And that's what becomes known as the primary net production. And it's available to consumers later on the food chain. In other words, the net primary production is equal to the energy that's associated with the gross primary production minus that energy that's lost by respiration. It's pretty simple, right? So another term you might come across or hear is called the standing crop. This is the total biomass of producers in one given area at any given time. So instinctively, you might be just thinking, well, is that not just the net primary productivity? The difference is that the net primary productivity is a rate, and the same value could be achieved by, for example, a highly productive or fast-growing plant, but in a small standing crop area, as it could be achieved by a less productive or slow-growing plant in a large standing crop area. There's also other variables that you have to consider here that affect standing crops, such as herbivory rates, and these can influence the standing crop by not necessarily affecting the net primary production. So effectively, the net primary production does not equal necessarily the high standing crop. Okay, now we've talked about how we can measure primary productivity. What you can do is pause this video for a moment to think about how it might be done. Okay, so hopefully you've thought a little bit about how that might be done. One of the first ideas you might have come to mind is actually just measuring changes in the biomass of the producer. And of course, this is a really great starting point, but there are a few limitations to this method, and hopefully you can get an idea of what they are by looking at this slide. Firstly, it's really difficult to measure biomass that's not easily seen, such as everything that's below the ground. And that biomass can be substantial and can result in an overestimation of the primary production of that crop. Secondly, the loss of biomass due to herbivory is another limitation that could be underestimating this time productivity. So a second method that's commonly used, particularly for terrestrial plants, is measuring carbon dioxide uptake and release in the chambers, such as the picture shown in here on your slide. And these chambers contain a highly sensitive carbon dioxide sensor. During photosynthesis, more CO2 is consumed than is produced by respiration. And this uptake can then represent an estimation of the net primary production. The same experimental setup can then be done in the absence of photosynthesis, in other words, in the dark, to measure the total respiration rate. And this value then can be added to the net primary production rate to give an estimation of the gross primary production in total. Now, these experiments are commonly used in ecology and are simply referred to as dark light bottle experiments. You can use a similar type of setup and increase the precision of these estimates using a similar setup based on performing and incubating the primary producers with isotopically labeled CO2, such as carbon-14 labeled, and then tracking the amount of that isotope as it's been incorporated into the biomass over a given time period. Another way you can measure productivity is to use the same type of dark light bottle experimental setup, but this time measure oxygen levels rather than CO2. This time, producers will release oxygen during photosynthesis and consume it during respiration. So in a bottle incubated in the light, the difference between the initial dissolved oxygen levels, and you can see that graphed in the graph here, at the end of the experiment, represents the net productivity. This same difference in the dark bottle represents the respiration rate. And the sum of both of these is the gross productivity of the system. So you might be wondering, what is the point? What is the benefit of measuring oxygen over carbon dioxide? Well, it really depends on the system you're studying. For example, oxygen measurements is the preferred way when you're measuring productivity in aquatic systems. And that's essentially because carbon dioxide is dissolved in water, can be rapidly then converted into bicarbonate. And so you don't get accurate measurements. Another measurement that's used to measure primary productivity is remote sensing. As the name suggests, this technique takes measurements from a distance using either satellites or aerial or drone images and captures large areas. So unlike the previous methods, this can be used to provide information of productivity at much larger spatial scales. These methods can work by looking at for example, the wavelength of light that is reflected or absorbed in an image, and then uses this information to estimate the standing crops the standing crop, of primary producers in a defined area. Changes in that predicted standing biomass over time can be used to estimate primary productivity. But as you might be thinking, an important note is that these methods, while they're powerful and have benefits to measure things in large scale, they have much more like the similar limitations we discussed earlier, is that they ultimately rely on changes in biomass that can be visualized above ground. So they don't take consideration that below ground biomass. Nevertheless, using these remote sensing techniques, one can get a picture of the primary production across the globe, similar to what you can see now in the slide here. In terrestrial systems, there's generally thought of as a higher level of primary productivity in the tropical regions, indicated here by the blued shading, and there's a decline towards the poles that's indicated here by the red shading. In marine systems, though, remote sensing of photosynthetic pigments predominantly being produced by phytoplankton in the water highlights that the majority of the primary production is occurring along the coastlines. And in particular, this is associated with higher around areas of river outflows and upwelling, due to these areas being relatively high source of otherwise limiting nitrogen, phosphorus nutrients. Now, an important thing to note on this graph, though, is that the levels of primary production are indicated here in the warmer shades and not the cooler shades as in the terrestrial example I showed in the other slide. So we talked about upwelling. So upwelling occurs predominantly in response to these coastal onshore winds that pushes the surface water away. And as you imagine, then in replacing that, you get the nutrient deep waters coming to the surface. And meanwhile then, at the bottom of the ocean, one tube worm says to another, "Do you think life exists on the surface of the ocean?" And then the other responds by saying, "Nah, well, there are no hydrothermal vents up there. So what would they use to get their energy from?" I love this particular cartoon, even if it's a little bit daggy. So these hydrothermal vents are located deep in the ocean floor, about at depths of up to 1 kilometre. There in this environment, the sea floor rifts allow for seawater to filter into the Earth's crust, and this results in the precipitation of elemental sulfur and other metals that then percolates up and creates these massive blooms or black smokers or plumes, is that they're called. Importantly, in these ecosystems and environments, they receive no sunlight from the surface waters and very minimal inorganic nutrient input that comes from that surface waters down. Yet, as that cartoon just described and highlighted, these environments are rich in biomass. There's microbial mats that cover almost every surface, giant invertebrates, giant crabs and clams, and also tube worms like shown in the photograph here. So the question is, how do these communities gain that energy from the vents? And the answer to this is that these environments are home to a second group of autotrophs. And those are the ones that obtain their energy from inorganic compounds rather than sunlight. These organisms are known as chemolithoautotrophs or chemolithotrophs. They're not the invertebrate animals that you've just seen in the cartoon or the slide here, but rather they are the bacteria that are found in and around the vent and also in a symbiotic relationship with those invertebrates. Again, such as the tube worm here. So let's take a closer look at that tube worm symbiosis. Here is a schematic showing a tube worm and the symbiotic bacteria that live inside these specialized structures in the body cavity. This is obviously not to scale. These bacteria perform sulfide oxidation. That is, they use the electrons from the hydrogen sulfide that's coming up from the Earth's crust in these black smokers in a redox reaction with the oxygen from the water to gain energy that is then used to fix carbon dioxide. That is via a regular carbon cycle, and that carbon dioxide is then fixed into glucose and shared with the host. Okay, now, and these bacteria that are essentially providing the foundation for the entire ecosystem in much the same way that you might think of plants that are supporting the forest. Now what I want to do is just pop back into one of our earlier lectures that we had on decomposition. And I'm doing this because it had occurred to me that some of you may be thinking that these are the same type of bacteria we talked about earlier when we talked about those microbes that were important for food decay, wood decay specifically, in the deep sea. If so, you'd be understandably mistaken. However, despite there being the apparent commonalities of sulfur cycling in the deep sea microbes, then we were speaking about the sulfur being reduced by bacteria. They are using the sulfate as an electron acceptor, and they produce hydrogen sulfide. In this case, our tube worm symbionts are using hydrogen sulfide. Okay, let's go back to our lecture. We've so far focused on autotrophs. The other form of energy and carbon capture comes from the heterotrophs. So the word heterotroph means other nourishing. And essentially, these organisms obtain their energy by consuming energy-rich organic carbon compounds that have come from other organisms, originally from autotrophs. And they include things like all the animals, a large number of microbes, and like the autotrophs, they can be further split down into how they obtain their energy. First of all, we have the heterotrophs that obtain their energy from light. But they still rely on organic matter for the carbon source. Those ones that have been most extensively studied include organisms such as the purple sulfur bacteria that can be found in sediments and aquatic habitats, including extreme habitats like hot springs in Yellowstone National Park. But there are now reports of animals even that can gain energy from sunlight using specialized pigments in their bodies. For example, this oriental hornet here, shown in the picture, has these yellow pigmented stripes on the abdomen. And these stripes act like a type of natural solar cell that can convert the light absorbed from the sun through the pigment into electricity. You might have read about other animals that can use sunlight for energy. Such, for example, as corals or some sea slugs. Many of these animals, though, are doing it by forming a symbiotic relationship with photoautotrophic algae. And so they wouldn't be considered classically as true photoheterotrophs themselves. So finally, there's the group like us, the chemoorganotrophs. They are essentially most animals, many microbes, and they use ready-made organic carbon both for their energy and carbon source. So this is essentially what results in our secondary productivity. And this takes us back to looking at the food pyramid again. Primary productivity feeds into the secondary productivity, and as you might well assume, then the amount of secondary productivity is limited by primary productivity. So this relationship can be seen in the graph here on below on this slide. And this shows the positive correlation between the net primary production on the x-axis with herbivore biomass on the y-axis. And this is across a variety of different terrestrial habitats. Secondary productivity is divided then further into the different trophic levels, as we spoke about earlier in our decomposition lectures. So a discussion on these trophic levels is where we're going to pick up our conversation in the next module. Until then, I wish you all a really great day or evening, depending on where and when you're watching this video. See you later.